I would like to thank the conference organisers for the opportunity to present this poster, Detecting Multiple SARS-CoV-2 Variants from Short-Read Sequencing Reads of Community Wastewater Samples. Identifying variants of SARS-CoV-2 has been critical in responding to the COVID-19 public health emergency. Emergence of new variants has anticipated surges in case rates of COVID-19 and can inform public health response. To date, most variant detection has been performed by sequencing the SARS-CoV-2 genome from individual patient samples, typically taken from positive PCR tests. This has enabled publication of over 5.7 million sequences in databases such as the NCBI's GenBank and in GISAID. This approach, however, has some deficiencies. There can be a significant time lag between infection and patients getting tested due to a delay between infection and onset of symptoms, availability of testing facilities, and time for test results to be generated. Additionally, a trend towards home testing has affected sample availability. SARS-CoV-2 is detectable in pre-treatment wastewater collected from wastewater treatment facilities. Since viral shedding occurs before symptom onset, wastewater samples provide a more up-to-date representation of SARS-CoV-2 than patient samples. These samples have been used to estimate case rates in various communities. However, since each sample represents a community of viruses from dozens or even hundreds of patients, it is less clear how to use these samples to identify variants. This work in progress seeks to use bioinformatic approaches to identify multiple variants from single pooled SARS-CoV-2 samples collected from wastewater. We collected and sequenced viral RNA from 22 untreated wastewater samples from 3 treatment plants and 2 student dormitories in West Virginia during January and February 2022. Sequencing reads were aligned to the reference SARS-CoV-2 genome. For each sample, we identified all polymorphic sites and constructed a graph as follows. For each polymorphic locus, we create nodes for each allele observed at that locus. For each sequencing read, we create an edge between any 2 nodes spanned by that read. So each node in the graph is represented by both a genomic location and a genotype at that location. And 2 nodes are connected if and only if there is a single sequencing read, meaning a single copy of the virus that contains the given genotypes at the 2 locations. The aim is to identify genotypes that only occur together in the pool of viruses. For each connected component of the graph, we removed nodes representing a genomic locus until the graph became disconnected. We will show a visualization of this process in the next slides. We term the resulting connected components discriminating mutation sets. For samples with non-trivial discriminating mutation sets, we counted the number of reads matching each set. We compared the genotype of the discriminating mutation sets with data from outbreak.info to assign variant types to each set. To visualize the construction of discriminating mutation sets, let's start with an example of a connected graph component from one wastewater sample. The loci in this component are in the S gene, which codes for the spike protein. For clarity, we've organized the graph with loci left to right. In this example, there are exactly 2 genotypes at each polymorphic site, but the same process works if there are loci with 3 or more genotypes. We select a locus and remove it with the aim of dividing the graph into 2 disconnected components. In our current implementation, we choose the locus with the most connections, most edges. There may be a better approach than this. We continue removing nodes representing a single locus, repeating until the graph becomes disconnected. Not every sample will result in a meaningful disconnected mutation set. In some cases, we just reduce to a single locus. Our samples were collected beginning just before the emergence of the Omicron variant in West Virginia. Of the 7 samples collected in the first half of January, 5 generated discriminating mutation sets. The 1 sample collected before this period, when Delta dominated, and the 14 collected after this period, when Omicron had become dominated, did not generate discriminating mutation sets. This suggests that the methodology may be effective at detecting times at which a new variant is emerging and competing with an existing dominating variant. We were able to count the number of reads mapping to each of the discriminating mutation sets generated for 5 samples collected in the first half of January. These indicate variation in the Delta-Omicron proportions. We currently do not have any validation of these proportions, but we'll discuss this momentarily. One of the samples had a discriminating mutation set which was not readily identifiable as Delta, though it consisted of only 3 loci and was thus not highly informative. This is ongoing work which is still in progress. Our immediate next steps are, firstly, to look for a more efficient method for selecting loci for removal in creating the discriminating mutation sets. We would aim to have larger discriminating mutation sets with a different method. Secondly, our facility has archived viral samples from individual patients, which we have sequenced and for which we have identified the SARS-CoV-2 variant. We plan to create pools of these at known proportions in order to validate the methodology. The sequencing and bioinformatics in this presentation was supported by a CDC Epidemiology and Laboratory Capacity Grant, which was administered by the West Virginia Department of Health and Human Resources. The Marshall University Bioinformatics Core is additionally funded by the West Virginia IDEA Network of Biomedical Research Excellence and the West Virginia Clinical and Translational Science Institute, which are both supported by the National Institute for General Medical Sciences at the National Institutes of Health.